Well, welcome everyone. Thank you for joining today for this webinar on virus characterization for cell and gene therapy with nanoflow cytometry, of course, using the NanoFCM platform. I'm Ben Peacock. I lead NanoFCM's research and analysis services, which utilize our platform for customers in both Europe and North America who are interested in characterizing LNPs, EVs, and of course, viruses. Today, we'll be sharing with you how our Flow NanoAnalyzer can help your research, control, and development efforts for viral products in cell and gene therapy. Today, Clayton Deighan is going to be leading this webinar. He's developed a broad expertise in nanoparticle detection and characterization technologies, and now leads our team in the United States. Throughout the webinar, if you've got any questions, feel free to put them in the Q&A box. We'll be getting to them live as soon as we're able to. And yeah, with that, I'll hand things over to Clayton.
Hi, everyone. Thanks, Ben, for the kind introduction. I'm really excited to share this presentation with everyone today, talk about some of the really cool things you can do with the Flow NanoAnalyzer for your virus samples. And it basically all comes down to really robust single particle interrogation. So without further ado, I'm going to jump right in. Here's our presentation outline. We'll spend a brief period of time going over the NanoFCM operational principles. And this is just so everyone who might not yet be familiar with what is different about the method by which we do this single nanoparticle analysis on the Flow NanoAnalyzer. We're going to cover that so everyone's on the same page about why it helps the virus user realize their sample in these complex solutions. Then we'll see examples of lentivirus analysis, adenovirus analysis, and HSV analysis. So let's go over these operational principles. The key characteristics of the Flow NanoAnalyzer, and maybe here, cytometry is a bit of a misnomer. The Flow NanoAnalyzer doesn't analyze cells. It produces flow cytometry-like data for nanoparticles. So our samples that are composed of viruses and the other components which might be riding along with them in our purification trains are perfect candidates for this type of nano flow cytometry analysis. And the characteristics that drive this are a very fine sample core stream. So the sample is injected through a capillary and then released into a larger flow cell in a very narrow stream. And at appropriate concentrations, the particles are going to line up single file and they'll pass through our lasers, which we're going to see the orientation of in a second, one at a time. And then they do this not just one at a time, but very slowly. The flow rate at which our instrument moves is orders of magnitude slower than other instruments. And that creates dwell time of the particles inside the laser beam or scattering or emission cross-section. And that gives them the time needed to scatter or emit the fluorescent signals that we need to perform this type of analysis. And we pair that up with very sensitive single photon counting APDs as the detector bodies, both in the scatter and fluorescent channels. And so you combine that sample core stream, slow flow rate, real single particle interrogation, and you end up with something that looks like we're going to see in a zoom into this video in the next slide, where we prepare a small amount of sample, something like 100 microliters diluted in a tube. We can make a lot less because the real consumption during a measurement is going to be about 1 microliter. And you'll set it on the elevator. The sample goes up and it's injected in the capillary. And the capillary at the other end terminates in a flow cell. And as the particles are released into the flow cell, they are passing through the lasers, and all the lasers and detectors are collinear. So there's no time component. There's no separation in space of when the particle interacts with different lasers or detectors. It all goes on down one line, which is very valuable for interpreting the nanoscale data here. And what that looks like is little peaks coming off, or what we're going to see on the next slide are event burst traces. And these represent the amount of light scattered by the particle in the top trace, and then its corresponding fluorescent emission intensity in the other two channels, which are configurable by the user. What this does is provide robust and sensitive data because we track in the different channels the event bursts occurring, and then look in the corresponding fluorescent, or sometimes we trigger on the fluorescence and look at the scatter. But here we see a nice example of, we'll think of this as a labeled virus. We have a protein on the virus we want to label. We have a labeled antibody, something very intuitive that looks just like flow cytometry, but scaled down here, we're going to let the single viruses pass through the laser, as shown in the schematic on the left. They're going to send their signals out to these single photon counting APDs, and the data we recover looks like the middle trace of these event burst traces. On the top, we have our side scatter. That's telling us that particles are passing by the detector, and we're going to learn how we talk about their size very shortly. But the bottom trace is the fluorescent detector at the same time, and at certain times, both of the detectors are triggering. We have a scattered particle that's showing us that it's fluorescent with our label. And at other times, we have a particle passing through the scatter zone that does not have the label. And then our data on the output of that looks a lot like what flow looks like. We have a positive population among all our particles for the marker of interest, and we have a negative population. And being able to interpret and interrogate different properties through this very simple display of the data as positives, negatives, how bright are they, how quantitative is that data, because we can confirm the particles are occurring one at a time through the burst triggering. This makes this really true particles. It makes the data robust, and it helps us quantify all these different properties we're going to look at and all these different intuitive ways that we can label the particles, as we're going to see. And to really make this go, what makes the NanoAnalyzer really work is our robust standards in our routine daily setup. So when we want to talk about having these unlabeled and labeled particles with distinct size, concentration, and phenotyping data, like we just saw, we're going to think of them as cells. We need to be sure that we have single particles. We need to be sure we know their size. We want to quantitate how many there are, and we want to quantitate their other features. So when we look at our total particle size distribution here, we have our size distribution. If you're a routine DLS user or NTA user, you're going to find that these size distributions themselves are largely confirmatory for data you already make with maybe more resolution. Maybe it shows a more refined picture of what the size distribution is instead of, say, the DLS giving you an output which is consistent but may not capture the accurate shape of the distribution. We also report the concentration of both the subsets that are labeled and the total. You don't have to do any labeling to recover the particle concentration, but we're going to see as we go down how determining the concentration of distinct subsets among all the particles that we can see in these type of samples is critical for knowing which sets of particles are actually important. Then we get the size information. So passing over the scatter detector, the height of the scattering is, of course, related to the size in the mixture, but knowing its true size requires us to provide strong calibration tools, which we do as these really narrowly mixed reference standards. So the 4-peak mix on the bottom is a representation of data you would make every single day when you set up the size calibration for the instrument. The beads are mixed in solution, and it absolutely does resolve them to that degree. The resolution on this instrument is sub 10 nanometers for peak to peak, even in mixtures, because they're truly being analyzed as singles. We've encountered other technologies that may struggle with swarming, may have times where multiple particles are being interrogated, and interrogating the one particle affects your ability to interrogate the other, or it provides a masking effect. Because we interrogate them as true singles, we achieve very high resolution based on the scattering heights for what the particle size is, and we confirm that all the time with our strong reference standards that give you real operational power all the way from 40 nanometers to a micron. And we have a similar set of beads that cover a bigger range too, that show you that it does have similar resolving power over the bigger size range, where sometimes other techniques work well at small, some work at big. Really, we work really well over our entire quoted size range here. For driving the concentration, we have a reference standard that we provide that is orthogonally verifiable with, say, a light scattering measurement on what is the particle concentration. You can confirm the number we put on it, and then we let that drive all of our concentration for the data we gather. And those two things, strong size standards, real counting reference standards, create this robust data for you. So let's get into some real analysis here of some actual samples we care about. And lentivirus is a big one. Let's start there. So what type of lentivirus samples are we readily analyzing, or where does the interest come from? The lentiviruses we mainly handle are related to CAR T therapies. I'm not going to sit here and give an expose on CAR T. We'll briefly visit that the lentiviruses in this space are used to transform T cells, which are harvested from individual patients, into CAR T cells for reintroduction into the patient to be a cancer therapy and go into their body and remove the cancer. In this environment, this lentivirus is something like an 80 to 120 nanometer particle. We're going to see that this inhomogeneity is real, and this creates clarity on why these are hard to analyze. They are not a distinct narrow picovirus. They have a range of sizes, and we're going to see there's some contaminants and co-isolating particles that will also impact the ability to analyze these particles. They are enveloped. They have a membrane. They have membrane proteins, and we're going to exploit that fact repeatedly here. They carry a single-stranded RNA genome of around 10 kb. We're also going to exploit that. We're going to interrogate the particles in the mixture for whether they contain RNA. And we have a little schematic here. We have our envelope. We're going to be able to label these different proteins. We're going to label VSVG a lot here. Inside, we have our RNA genome and a capsid protein P24, which we're also going to label here. And really, the virus is a very important part of the CAR T therapy process. They're an expensive component. Up to 30% of the cost of the therapy is actually creating, retrieving, purifying, having the virus ready to use and do its transformation. And obviously, it has a direct impact on the quality of each CAR T therapy. And thus, they require drug substance level quality control. The lentivirus for these therapies is considered a drug substance. And that is hard to deal with at times because, in fact, when these viruses are produced in solution, we receive back from the cells doing the work a whole range of different particles, not to mention extracellular vesicles, which we're going to see consistently throughout the data here. But this is just among the types of things that the cells can make when they're trying to make a lentivirus. We can get something like the left panel. We can get a functional virion. It has a genome, it has its capsid proteins, it's properly enveloped, and it has its recognition factors on the surface like VSVG. We can also end up with any of the range of 4 things we can think of as non-functional here. We can have non-functional virions which lack their genome, but may contain all the other characteristics they need. That it's a 100 nanometer particle. It has VSVG, but it doesn't have a genome. Or we can have the opposite situation. We can have things that look like 100 nanometer particles that have RNA, but they don't have the VSVG on them, and they're not going to be very functional. And then, as we all already know, P24 is prevalent in solution when these are being made by cells, and free genomes or cell-free nucleic acids are going to be another factor that comes out here. So being able to hone in on which particles contain multiple components is going to be critical to sorting out among a complex mixture which of these are actually whole virions. And how does this lend itself into the virus QC? We're talking about the size of the particles. We're talking about the concentration in particles per mil of the particles. We're going to see how purity becomes apparent when we analyze a set of particles and we're able to say this percentage of the particles fall into this category. We're going to talk about the degree of protein labeling between different preps. And we're going to talk about stability and the correlation with some of your other assays. So in development, screening of stable and high-producing cell lines can be achieved without needing to purify the virus deeply. We're going to see how these things can be detected throughout the purification train. That allows you to optimize plasmid transfection, choose a mode of culture, a type of cell media, without needing to run expensive purification processes. Same concept in manufacturing. Because we can label and identify the subset of interest in complex solutions, we don't need extensive purification to evaluate the performance of different unit operations. So we can talk about optimizing types of chromatography resins, parameters of membrane performance, choices of buffers, determining operating conditions, and monitoring the effects and quantifying them on virus recovery, particle size, and purity relative to their proposed performance within the process. And in quality control, assessing the final purity, identity, safety, and potency of the LVs can be achieved with different types of correlative assays and the strategies that we're going to see in the next few slides. Monitoring the stability of LVs for storage and transportation with this very deep interrogation, and tracking consistency across different batches, which we'll see a nice comparison of. So where then does the flow NanoAnalyzer fit into the process? Is it uniquely for quality control? Is it for polished products? Actually, no. We're going to find here that everywhere we've put a green arrow in our right virus-based therapeutics is a place where you can grab a small sample from and perform any of the types of analysis that we're going to show in the next few slides. You can get at the labeling of the distinct viruses. You can size them distinctly. You can see the way that the RNA intercalating dyes intercalate the different sets of particles. And you don't need them to be highly polished from potential contaminants like we saw in the fragment slide with the incomplete virion type particles, and also the main contaminating component, the extracellular vesicles, which the cells bud off. So we've talked about the types of analysis we're going to look at. We're going to get size and concentration. We see an example in the upper left here of a big peak near 85, 90 nanometers in this lentivirus laden sample, and small peak out to the left. And we're going to learn a lot of different things about why do we see these other particles present in something we think should be really just laden with viruses. Very strong dilution linearity for this type of method, including within the subsets. And that is, again, driven by our good standards that you use to start out. Using the good standards creates good linearity because you're operating in a range where the instrument does really well. We're going to find this relationship between size and function. These lentiviral particles are often with other similarly sized particles, but we're going to find that our co-isolated particles, those which display the multiple components we need to be active, are going to be uniquely sized relative to the greater range that we'll see in that looks typical of many cell culture experiments. And then repeatability here of concentration, this links back to the standards. By using routine standards, we achieve really nice reproducibility of our concentration measurements across different batches, days, etc. We'll start at something simple, and we'll take here a lentivirus sample, and we'll perform 3 different types of single color staining. And we've labeled them across the top as the membrane. So you can think of this staining as a general membrane dye. I have a bunch of particles in solution. Do they have membranes on them? And you can exploit different types of your favorite types of membrane dyes. We will often use some called memglow that are specific for lipid bilayers. You can use any of the typical membrane intercalators like the DIL series or the cell mask series and determine among my particles, these have membranes. And sure enough, when you look at this type of sample, you find, okay, at least 90% of these particles are membranous. And that's maybe trivial and still might not give you enough information to determine, okay, if I have vesicles, does looking at the membrane tell me that something's a virus? Actually, it really doesn't. Those are going to look very similar. So then we do a more virus-specific staining with P24, the capsid protein. And here we've now find that, not 100% of the particles are carrying P24, only 55% are. And then when we do our RNA stain, and this is with something like the Cyto series of dyes in the right panel, we're back up to a very high percentage of the particles containing RNA. And this may seem counterintuitive because we say, well, we have our P24. Do we have other particles that have genomes and not P24? In practice here, a lot of the extracellular vesicle components, which are not viruses, but are from the cells, they're known to contain RNA too. So we're going to need to bring together some combination of these different single color stains in order to learn enough about the distinct subsets of particles to start identifying which ones are really the virions of interest here, even though we can do any of these things individually. And when we do that, and we'll show it here with a surface stain, so this will be protein on the surface plus the nucleic acid stain. Now we're able to start to see among these dual stainers are really the ones that we're looking for. So we have our schematic on the left. We've talked about the capsid envelope, the RNA genome, and now the recognition marker VSVG. So we've used anti-VSVG in this figure. This is slightly different in the dot plot in the middle from the previous slide. On the previous slide, we were looking at the scatter versus the single color stain. Here, all of these particles have a scatter data point, but we're actually looking at their co-staining of, does the particle have VSVG on its surface? And does it also have nucleic acid inside? And now we see in population 1, this set of, on the right, the size distribution for covers right over that 80 to 100 nanometer range in these complete virions. And then if we look at the other interesting little populations, P2 is kind of interesting. That'd be VSVG particles that don't have RNA. This is actually not too uncommon because the VSVG as an envelope protein or membrane protein can be displayed on EVs. And we see that their size distribution in red on the right is quite small. And then our P3, these are our RNA carriers that don't have VSVG. And we find out once again in the right, in the green size distribution, these look more like EVs. These are smaller still than most of our intact viruses, and they don't have VSVG on their surface, although they contain some type of nucleic acid. And then we have P4. These are our non-staining particles. We saw them as like 10% in the membrane stain. And a lot of people would term these process-related particles, especially as we move through a separation strain. These are particles which can be identified, can be quantified, can be tracked. Their identity is not easily known based on the staining methods we're going to use here. So process-related particles we're able to put our finger on, quantify, and quantify our active particles in here too. So we have a couple other configurations for active particles. And what are we going to find out here? Over here, we had our complete virions. Let's look a little bit closer at the number. It's 45% of our complete virions in here. That's VSVG and nucleic acid and size distribution through this lens. We can look at this through a couple other lenses, though. We could say, let's talk about P24, which we saw before, and membrane localization. And that plays back into the first single color stain, but now we're overlapping the two. So in the right half on the membrane component, we see we've got very high percentage. We're back up at 92% of the labeling's membrane. Almost all of our P24 is in the upper right population there, meaning that the P24 particles have membranes on them. And this is fairly intuitive, but we're now confirming, okay, that gives us 45% again. So we've got another good track on 45% of these particles are looking like viruses. When we combine the P24 with VSVG, P1 again being important, we want to see that we have the VSVG and P24 in the same particle. Again, 40% of the particles, the same type of number that we're seeing anytime we're overlaying the two types of staining, comes back to us. And lastly, nucleic acid plus P24, again, the same 42% that we saw, or 45% we saw with VSVG plus nucleic acid, we're finding overlaid in the P24 and nucleic acid too. So these different components, which can be the routine indicator for your quality controller, which is linked to your activity, that's exactly what you can determine by running a correlative study that we'll show an example of later. We'll stop here and compare some products. So here we have two different products. So what type of quantification are we able to make in? We have our two single color stains. We have our nucleic acid stain, which again, we see, okay, product B has some more particles with nucleic acids in them between the top and bottom of the two left panels. But we learned before that that's not quite enough to know whether those are EVs that are riding along from a separation. Is it actually differences in virus? Our VSVG staining definitely looks a bit different. We've got a much higher percentage of the particles in product B, which have VSVG, but they're also much dimmer. So the positive rate is high, but the intensity per particle is low. And then in the top figure, we have a lower rate of positivity, but it's intense per particle. The copy number of VSVG on each particle is high. And then in our combination of nucleic acid and VSVG, we obviously have high nucleic acid localization. So we see in both cases, the VSVG loaded particles are largely full of nucleic acids. And thus we see on the right, almost all of our VSVG stains become positive, but we still only have high differences in the co-localization of the two, because originally the VSVG in the top panel only covered 46% of the particles. And when we look at the numerical values for these, we recover things like between these two product runs, the difference in the total particle concentration, the difference in the total particle size distributions we see. The VSVG plus particle size is actually changing a little bit. They're a little bit bigger in product B, where they have a very high positive ratio, but then we can see in their medium fluorescence intensity that there's actually less VSVG per particle in product B. And which of these is the better product is exactly what people are determining by linking that to their downstream assays, where you would determine the transducing units. You now have a range of different total particles, nucleic acid particles, VSVGs, nucleic acid plus VSVGs, like we've determined, that can be compared to that. I think I want to do these in this order. I'm going to go back to the previous one. And then when you get into this process monitoring, because you can follow this along the whole process and you can grab these samples from throughout the separation string, you can now link up your NanoFCM determined particles of interest, whether you go with the VSVG membrane route or the VSVG nucleic acid route. You can link up your perception of complete virions with your transduction titer. And now you're starting to learn at earlier stages, especially among many batches or new batches, whether they're comparable to old batches that have known transduction titers. And you can start to make earlier decisions about how to handle the material and whether it's going to be useful downstream, or if you're doing kind of the different things we talked about in development, you can screen those things earlier without doing steps 1 through 6. You can screen them at step 1 and 2 and have this link back to the activity that's become known from the other correlative experiment. Let me go back one slide. So overall, when we look at those methods of comparison, we have our viral particles, and then we have our methods on the left. The transducing unit, it's the gold standard. We're always going to be comparing to it. I think it'll be important to conduct correlative studies with your transduction assay. Difference is this data is coming back to you from a sample in a couple of minutes, like less than 5. And the transduction assay can take obviously a couple of days. And there you're going to determine your functional viruses, and that's going to be our standard for how many functional viruses relative to the other samples that we're looking at. Can't quantify much else with that assay, but obviously we're not going to get away from doing that. And then in our P24 ELISA, we can quantify lots of different things in addition to P24, but it's hard to determine which are which. And then the concentration of total particles, which we're determining here with the flow NanoAnalyzer, or the concentration of complete lentiviruses, can both be determined and kind of separated from the immature, broken, or insufficient envelope protein, empty particle-like things that are in that mix with the active particles. And that's all enabled by the fluorescent labeling and its sensitivity, and the sensitivity and resolution of the scatter. So as we hit on, this has numerous applications across the development, manufacturing, and quality control of these viruses, or what could be summed up as virus QC for these drug substances like lentiviruses. And we're happy to discuss this further with anyone who wants to have a conversation, learn more about it. If you're making lentiviruses and this looks interesting, reach out to us at the conclusion of this, and we'll be happy to set you up with someone from the team to discuss more about your particular application. And with that, we're going to jump on to adenovirus, something a little different. So the lentivirus are these quite complex enveloped viruses. They come in many flavors. Adenos tend to be a little bit more binary, and they come out as intact and full of their genome or empty capsids. And what we'll see here is that we can determine with adenoviruses their kind of mixture of empty fulls just based on their scattering signature. So intact EDVs and their empty capsids have the same particle size, but due to the deletion of the genome, the empty capsids have a lower refractive index in this application. And we can take advantage of that in a mixture where we know we have adenoviruses, and sometimes these are easier to purify being on envelope, so you can get relatively clean solutions of just adenos. And we can see them differentially. And when we do a nice mixture of intacts and empties that have been separately purified and remixed together in different ratios ranging from 1 to 0 to 0 to 1, and the thirds in between and 50-50, we're able to recover a good linear correlation with the theoretical complete virus proportion and the actual measured proportion. And again, these will stand out from EVs too by their scattering signature, so you can collect them in those more complex solutions that you may have trouble with bulk technologies or other things that don't have labeling capabilities, seeing the viruses among the many other particles that can be present. Our final example for today, a very interesting one, and another just a really good testament to the fact that these types of samples are complex. When we've made virus in solution, very rarely will we get something that looks like a standard, let's say, in its narrowness for how narrow the particles are. So another virus is a little bit bigger. So our composition and structure of the HSV, we've got our envelope proteins, the outer tegument, the integument, major capsid proteins. This is still another step up in complexity from the lentivirus. And the HSV is large. It's 152 kb. So our lentivirus is around 10 kb. This is tenfold larger, 152 kb double-stranded DNA virus. Diameter is known to be 150 to 200 nanometers, divided into two serotypes, HSV1, HSV2. We've seen there are some HSV products on the market as therapies. The outermost layer is a lipid bilayer, so this is enveloped again, coated with at least 12 different envelope proteins, of which GB, GD, and GE are going to be of interest here, and that's what we're going to look at in terms of our staining with antibodies on these HSV-type particles. So we look at the size and concentration measurement. We can get a nice linear dilution of these, but when we recover the size distribution, we've once again encountered, kind of like the lentivirus, we don't have that distinct peak that says, oh, it's very easy to see the virus. We've got bigger particles in here. We can see some larger things between 100 and 150, but there's not jumping out at us that sizing these alone is enough to determine that they're exactly what we think they are in this complex mixture. So we go to our usual suspects, the membrane and nucleic acid dyes. And once again, we found, hey, a lot of these particles that co-isolate or are riding along with our HSV, they've got membranes and nucleic acids on them. So we're, again, going to need a little bit more information to draw out among this complex mixture, which ones are the ones I'm really looking for. And we start to use GB, GD, and GE. And now we're able to put our finger on where the viruses are. And we notice, too, the skew along the right axis of all of these markers are skewed to the right side of our distribution. They, in fact, are in the bigger particles. And it's likely we have something like extracellular vesicles floating around in the small side, which obviously, as we saw in the previous slide, are membranous, and they can entrain nucleic acids. So the positive rates of these key infectious proteins here among all the particles that are in this isolate for GB was 20%, GD is 40% or 49.2%, and GE was 25% respectively. And these positive particles, we said, we get that confirmatory bit that these are all in the bigger particles in the mixture. These are the viral particles, and these are not as spread out on what might be the EV particles riding along. And in that vein, we've also done some analysis here of the exosome fraction, and I really like the middle chart here. We can tell the tetraspanins being very promiscuous membrane proteins anyway. They're obviously spread out across a number of these particles, but our CD63, a classical EV exosome marker, we can see is concentrated to the small side now, opposite the G proteins that we looked at And it is actually around the small side of the distribution where we might think that the extracellular vesicles that we're interrogating are concentrated. And the particle proportions here are similar to our virus particles. They're in the 10, 30, 10% range for these common tetraspanin markers. They're easy to detect and label. And we find out that they are spread out across, in fact, some of the viral particles too, and that some of them are concentrated in the smaller ones, differentially from our G proteins, which are concentrated in our known bigger viral particles. And the result here really indicates that these exosomes are a common impurity in HSV samples, and I think the same could be said for a lot of different virus samples, particularly that are enveloped. It is fundamentally difficult to remove EVs thoroughly from an enveloped virus sample, and now you have an opportunity to quantitate, capture, know what fraction of them are present, whether a separation strategy is having any impact on them, and if there's anything that you can add to further characterize them or know what their effect is in different types of preps. When we bring in the colocalization of nucleic acid with back to our key infectious proteins, we're now able to see that the proportion of nucleic acid, and this is vertical axis in the chart is going to be the G proteins. We see that virtually all of our G protein positive particles are positive for nucleic acids, which may be intuitive because they have large genomes. So we're going to see them fairly easily. All of those particles, which are GD or GE positive, return the same percentage positive among the total because we show nearly complete labeling of the GD or GE particles with the nucleic acid dye here. So in summary, we visited 3 nice examples of how the flow NanoAnalyzer can characterize viruses across your product and process development trains. And it's doing so by bringing you real concentration that's driven by a standard for the total and subpopulations of the particles that you'll interrogate. It's doing so with high resolution size distribution measurements, which allow us to exploit the high resolution sizing coupled with multiple labeling to reveal distinct subsets of particles in what look like largely continuous distributions when they're just analyzed by size. The broad characterization we've provided includes proteins, lipids, and nucleic acids through the fluorescence detection, and we're always providing that really high resolution scatter detection coupled with that and seeing those interesting subsets. And then speed, we're collecting this data in real 4 to 5 minutes per sample. You place the sample on, inject for 45 seconds, collect a minute of data, remove the sample. And when you're doing something you've done routinely, you can often process your data to completion in the time that you'll run your 1-minute cleaning before your next sample is ready to inject. So all this is making nanoflow cytometry from NanoFCM a powerful and flexible platform for comprehensive nanoparticle, and in this case, virus analysis. And we will say thank you